Okay, so let's move to the next talk. Next talk will be given by Roy Weinstain. Roy Weinstain from the Faculty of Life Science, Department of Molecular Biology and Ecology of Plants. And the talk will be in vivo targeting of hydrogen peroxide with activable cell-penetrating peptides.
Right, thank you very much. Thank you for the opportunity to be here. I'm a new faculty member in the Life Sciences faculty in Tel Aviv, and the work that I'm going to talk about today was mainly done during my postdoc, and we are starting to continue it now. So it's very different from the previous talk in its nature, and mainly we are interested in vivo targeting of hydrogen peroxide, as you can see from the topic. And hydrogen peroxide is a member, this molecule is a member of a larger family of molecules which are termed reactive oxygen species. This is a family of very small and very reactive molecules that is being generated in organisms in our bodies all the time as a part of normal processes, and the body has ways to get rid of this kind of molecule that can cause a lot of biological damage. It was identified that a lot of reactive oxygen species are overproduced or overabundant in many pathological conditions. Now, there's a very big debate today in science whether reactive oxygen species are causes or drivers of many of these pathologies, or they are just a symptom or an outcome of such kind of pathologies. I'm not going to dive into this very hot debate, but our work is based on the fact that it is recognized that there are a lot of reactive oxygen species, and especially hydrogen peroxide, in many of these pathologies. Now, why is it important? It's important because if they are overabundant, we can use them as a target to target imaging agent and drugs to oxidative stress-related diseases, as the diseases that you see here on the slide. So, in order to do targeting of hydrogen peroxide for imaging of diseases or for drug delivery, one needs to come up with a targeting mechanism in order to find the hydrogen peroxide in vivo. And according to the three questions that were mentioned here before, so currently the state of the art is that there are no targeting mechanism for hydrogen peroxide which are suitable for in vivo application. And this is what we set ourselves to do. And the mechanism that we wanted to use is a general mechanism that was developed in Professor Roger Chen's lab several years ago, which is termed activatable cell-penetrating peptides. The mechanism is based on a cell-penetrating peptide, and there are various sequences of this kind of peptides. Usually they are positively charged peptides. Our lab uses a series of 9 arginines, but there are other sequences. And this kind of peptides have the ability to interact with biological membranes, which are negatively charged, and penetrate through them inside the cells. Now, it was also found that many of these peptides can carry cargo, and this kind of cargo can be small molecules, it can be proteins, it can even be nanoparticles and liposomes, and they can still interact with membranes and penetrate into cells with their cargo on them. Now, the problem with cell-penetrating peptides is that they are not specific. So they would interact with any membrane that they see first and penetrate into that cell. So if you introduce a cell-penetrating peptide with a drug into an organism, it will just interact with the first cells, and it will not have any specificity. It's usually a series of between 6 and 12 amino acids. So they're quite small. To confer specificity to cell-penetrating peptide, our lab has developed a mechanism in which you connect the cell-penetrating peptide through a linker to a series of, or to a small inhibitory peptide which is negatively charged. In its intact form, this molecule forms a hairpin structure where the negative charges countering the positive charges, and now they cannot interact with cell membranes and they cannot penetrate into cells. However, in the presence of a cleaving agent, something that can cut the linker, the two peptides can separate and dissociate from one another, and now the cell-penetrating peptide can now interact with cellular membranes and penetrate into them. So our lab has developed a lot of this targeting mechanism to target enzymes by placing specific sequences for enzymes as a linker. And we were using this kind of devices to image the activity of a lot of matrix metalloproteases, which are overabundant in cancer, to do cancer imaging by fluorescence, by MRI, and by PET, and we also use it for drug delivery. We can also target it to different enzymes like thrombin in order to image sclerotic plaques, and even to classify them as and even to use them for classification. So we wanted to take this kind of mechanism and use it in order to target hydrogen peroxide, and I'll just show you why we chose specifically hydrogen peroxide from all the reactive oxygen species. So if you look at the half-life of most of the physiologically relevant reactive oxygen species, they are usually very, very reactive, so they have very short lifetime. So when they are being generated inside the cell, they cannot go outside the cells. And I'm sorry, as I showed you before, the ACPPs, the activatable cell-penetrating peptides, are designed to be activated outside the cells and only then penetrate inside. So we need the activating agent to be present outside the cells. So from all the physiologically relevant reactive oxygen species, hydrogen peroxide is almost the only one that has sufficient lifetime in order to penetrate, in order to diffuse outside the cells. Also, because it's not a charged species and it's not a radical, it can easily diffuse through biological membranes. In addition, the oxidative environment that is present in the extracellular matrix makes the concentration of hydrogen peroxide outside the cells about 7 to 10-fold higher than what it is inside the cells. So all these settings makes hydrogen peroxide a very approachable target for our ACPPs. Now, if we want to target hydrogen peroxide, it will be useful to understand what are the concentration of physiological hydrogen peroxide, and there isn't a lot of data about physiological concentration of hydrogen peroxide in vivo. It's very difficult to measure, but from our scan on the literature, what we can find is that this value of about 2 to 4 micromolar is what usually seem to be the value of hydrogen peroxide in normal conditions, and in various pathological conditions you can see an increase of about at least 10-fold of hydrogen peroxide concentrations. So these are the physiological concentrations that we are aiming to be that our system will be sensitive enough if we want to target hydrogen peroxide in vivo. And in order to do the targeting, we needed to design a linker that will be able to detect hydrogen peroxide and then cause the breakdown of the peptide in its presence. So for the sensing of hydrogen peroxide, we chose the reaction of boronic acids and boronates with hydrogen peroxide, which are being oxidized and generate a phenol. To confer the breakdown, or to facilitate the breakdown of the peptide, we want to use the reaction of what happens to a good leaving group when it's conjugated to a phenol on a benzylic position. And what happens is a spontaneous and rapid 1,6 elimination reaction, which causes the leaving group to leave the system. We thought to marry these two reactions together to generate a molecule that looks like this, which can sense hydrogen peroxide and also have a very good leaving group, a carbamic acid in this position, which should be able to fall apart. And this is how we envision the system to look like. So we have the cell-penetrating peptide, a series of 9 arginine, connected through the hydrogen peroxide-sensitive linker to the inhibitory peptide. In the presence of hydrogen peroxide, the boronic acid should oxidize into a phenol, and then a 1,6 elimination reaction should cause the defragmentation of the peptide and release the cell-penetrating peptide to interact with cellular membranes. And in order to visualize the process and to study the system, we thought that we will label both parts of the ACPP with fluorescent molecule. Because the molecule forms this hairpin structure, these two fluorophores are being placed very close to one another so they can form a fret pair. In a FRET pair, you can illuminate the first fluorophore, and instead of it emitting light, it transfers the energy to the second fluorophore, and this one will now do the light emission. And so after a reaction with hydrogen peroxide and the breakdown of the peptide, these two peptides will move further away from each other, and because FRET is very sensitive to distance, now when we excite the first fluorophore, we will see the emission coming out of it. So this is the beast that we eventually synthesized. What you see here is the series of 9 arginines connected through the self-cumulative linker, or the hydrogen peroxide sensitive linker, to a series of 9 glutamates, which are serving as the inhibitory peptide. And both parts are labeled with fluorophores. The cell-penetrating part is labeled with fluorescein, and the inhibitory part is labeled with Psi5. When we measure the spectrum of the fluorescence emission spectrum of this molecule, when we excite it at the fluorescein wavelengths at 488 nanometers, what we can see is that we hardly get any fluorescence coming out of the fluorescein. All of the re-emission that we are getting is coming from the Psi5 fluorophore. However, when we treat the molecule with hydrogen peroxide, you can see that now we lose all the re-emission from the Psi5 and we regain the emission of the original fluorescein. To make sure that this is not just a specific phenomena for the fluorescein Psi5 pair, we also labeled the peptide with 2 other different fluorophore, Alexa 488 and Alexa 594. And here you can see when the molecule is intact, we see when we excite the Alexa 488, we get the re-emission from Alexa 594. When we treat it with hydrogen peroxide, we can see the FRET changes. And monitoring the FRET change will enable us to monitor the reaction with hydrogen peroxide. To make sure that the molecule indeed falls apart when we treat it with hydrogen peroxide, we run it on an SDS page gel. And you can see here, when the molecule is intact, we see a FRET band. And when we cleave the molecule and treat it with hydrogen peroxide, this band completely disappears. We see a shorter band, which means the molecule is smaller, and all we get is this emission from the fluorescein, so it means that the molecule indeed falls apart. To make sure that it falls apart because of the reaction with hydrogen peroxide, we synthesized, this is the ACPP that we are working with, we synthesized another one that instead of the hydrogen peroxide sensitive linker, now we have just a PEG-6 linker. This should not be sensitive to hydrogen peroxide, and indeed when we treat both molecules with hydrogen peroxide, in one of them we see an increase in the FRET change, and on the other one, which should be insensitive, we don't see any changes in the FRET. And so next we wanted to check or to test the sensitivity and the specificity of our probe to hydrogen peroxide. So in regard to sensitivity, we took the probe and put it in a tube and started adding hydrogen peroxide in increasing concentrations. And what we saw is that we can monitor the FRET change, and you can see that it is linearly dependent on the concentration of hydrogen peroxide. And the detection limit that we can achieve is somewhere below 10 micromolar, which makes us pretty good in the physiological range of what we were seeing with hydrogen peroxide from the literature. In regard to specificity, we treated the probe with various physiologically relevant reactive oxygen species, and we confirmed that the probe is indeed pretty specific to hydrogen peroxide. Next, we want to see if the molecule retains its ability to report on hydrogen peroxide in cellular environment. So first we took HL60 promyolytic cells and added the probe and just started to add exogenously hydrogen peroxide at increasing concentrations. And we can see that we can monitor the changes in FRET. It is still linear. Our detection limit is still below 10 micromolar. So we can do that in cellular environment. So the next question was, can we measure endogenously produced hydrogen peroxide? For that, HL60 cells can be activated with Forbol myristate acetate, or in short PMA. When you treat these promyolytic cells with PMA, they undergo some transformation that causes them to undergo an oxidative burst, a process called oxidative burst, in which they are generating a lot of reactive oxygen species, including hydrogen peroxide. And what we could see there is that when we treat cells with PMA, we can measure or monitor the changes in FRET, and because we have the calibration curve from the data before, we can calculate that this kind of cells generate about 1 nanomole of hydrogen peroxide per 10,000 cells per hour. In addition, we could see that cells that even before they have been activated with PM8, they can generate a small amount of hydrogen peroxide, and we could calculate that this increase here is about 20-fold when they are undergoing oxidative burst. So we can image reactive oxygen species that are endogenously produced by cells. The next step was to see if we can measure reactive or monitor reactive hydrogen peroxide, that are being endogenously produced inside living organisms. And for that, we used a model in mice, or a model of lung inflammation in mice, which is a model that you can induce by challenging the mice with lipopolysaccharide, with LPS, intranasally introduced into the mice. And we can confirm the occurrence of inflammation by H&E staining, and can see here recruitment of macrophages and neutrophils into the areas of airways. So with this model, we turn to test our FRET hydrogen peroxide ACPPs. We introduced them intranasally, the similar way as we did the LPS, and we tested both kind of mice, the one that were challenged with LPS and the one that didn't, and we do a FRET imaging basically of the lungs, and we can see that there is an increase in the FRET in mice that were challenged with LPS. To make sure that this is a result of a reaction with hydrogen peroxide, we also treated LPS-challenged mice with penicillamine, or D-penicillamine, important to say. And D-penicillamines are scavengers of reactive oxygen species, including hydrogen peroxide. And you can see when we do the treatment with penicillamine, we lose all the FRET chains that we observed before. We did the same, we repeated the same experiment with the Alexa 488 and Alexa 594 version of our ACPPs, and here again we can see an increase in the FRET when we challenge the mice with LPS, which disappears when we add the hydrogen peroxide scavenger. We can quantify this data, and we can see that there is about twofold increase when we challenge the mice with LPS, and this completely disappears when we add the hydrogen peroxide scavenger. To make sure that the changes in FRET that we see here are indeed because the molecule fell apart and not because any other damage, biological damage to the molecule, we harvested the mice lungs, we homogenized them and ran them on a gel, and what we saw is usually observed only two fluorescent bands, one of them which has this spectrum which corresponds to the intact ACPP, meaning some ACPP that was not cleaved, and the other band which corresponds to this fluorescent spectrum which corresponds to our cell-penetrating peptide that was released from the system. And the same is true for the other version of the molecule. So at this point, our future directions with this project is we want to improve the kinetics of the reaction. And the importance of improving the kinetics is not only we'll be able to visualize the hydrogen peroxide quicker, because in the experiments that I showed you here, it took several hours to generate enough signal to observe the hydrogen peroxide. So improving the kinetics would improve the time frame of the experiment. It will also lower the detection limit of the reaction, of the probe. In addition, we want to move on to use better fluorophores for in vivo imaging, and we of course want to apply it for additional animal models. And I want to use the time that was remaining to show you how we start addressing improving the kinetics of the reaction. So to do that, we look back at the basic chemistry of what we are doing here, and it's all ACPP with all the fluorophores and all this complex system. Everything is based on this very simple reaction of the hydrogen peroxide with boronic acid. Mechanistically, this reaction is proceeding through several steps. The first step is a reaction of the boron with hydrogen peroxide. Then there's a migration of the ring from the boron to the oxygen, and then simple hydrolysis, which yields the phenol. And when you want to improve the kinetics of these kinds of reactions, you usually want to introduce some chemical modification that will influence the electronic state of the reaction. So in this case, or the problem in this mechanism, is that we have two steps here that requires opposite electronic states. So the first step of the reaction requires electron pouring or the more poor, the poorer the ring will be, the quicker the reaction will be. But the second step of the reaction requires electron donation or electron-rich rings to make it faster. And indeed, when this reaction was comprehensively studied during the 1950s and 1960s, it was discovered that no matter what kind of modification you made on the ring, make it richer, electron-richer or poorer, it didn't change the kinetics of the reaction to a significant extent. However, when we looked further into the details of how these experiments were done, they were all done under very acidic pH between 3.5 and 5. And we thought that maybe under physiological conditions, one step of the reaction will be more rate limiting than the other, which will open the way to expediting the reaction. And interestingly, no one has ever challenged this assumption that this reaction cannot be modified, or the kinetics of the reaction cannot be modified by changing the electronic state of the benzene ring, because everybody relied on the data that is presented here. So to test this, what we did is we took a boronate, which works as well as a boronic acid, and we introduced various electron donating or electron withdrawing groups in the para position, and we measured the rate of the reaction with hydrogen peroxide. And what we found is that actually you can, under physiological condition, enhance the rate of the reaction when you increase the electron density of the ring up to about 5-fold if you put a very strong electron donating group. And this translates very nicely into a Hammett plot. And what this mechanistically, from the chemical perspective, what it means is that under physiological condition, this step of the reaction is the rate limiting step, and we can influence it and make the reaction quicker, but by making the ring more electron rich. We also looked at what happens when we introduce modification at the ortho position. Here the picture is more complicated because in addition to the electronic consideration, there are also steric considerations, but still you can see that making the ring more electron rich usually makes the reaction quicker. And based on that, we have designed a new linker, which makes use of all of the insights that we've learned. And we think that with the new linker, which we started to synthesize, we can get about 10 to 15-fold increase in the reaction kinetics, and which should also translate into the lower detection limit. And this should enable us to emit hydrogen peroxide even in non-pathological conditions in vivo and in very reasonable time frames of about half an hour to an hour. And so this is pretty much where I thought to end the talk, unless I have a few more minutes. No. Okay, no, so I thought to introduce another concept, but I guess this will be enough. So I'll just finish here. I'll thank the lab members of Professor Chen's lab that were involved in this project, my current lab members, and the funding that was used in order to finance this experiment. And thank you very much for your attention.
Let's have some questions.
Thanks for the talk. One question, the cleavage is done outside the cells.
Yes.
So the cargo can go into a neighboring cell after the cleavage. It doesn't have to go into the oxygen peroxide producing cell.
You're right. This system is not cell-specific, but it's very specific to the area, because once the cell-penetrating peptide is released, it's very quickly interact with membranes, so it will stay in the very close vicinity to where it was activated. It will not be necessarily the exact cells that produce the hydrogen peroxide, but it will be in the same location. So we can retain a spatial resolution, but we can't say where the hydrogen peroxide came from. That's true.
Any more questions? Okay, so let's thank the speaker again.